Good morning, everyone. Good morning. I'm sorry to break up the conversation. It's very animated and good energy in here this morning. It's wonderful to see so many people, both familiar faces and new, for the fourth event in this Spotlight series. My name is Bod Buckby. I am a trustee and a patient here at the Christie, and it's my pleasure to host today's event. So everybody in this room and everybody in the Christie really understands how precious time is. So we really do value the hour that you're all choosing to spend here with us this morning, especially when you're giving up a beautiful sunny summer's day out there and you could all be enjoying that sunshine and being in the garden, catching up with jobs around the house. So we really do appreciate the time that you're giving. I hope that we can repay that. I think we've got a really fantastic set of speakers for today's event who's going to give you a little bit of an insight into some of the incredible work that's being done here at the Christie to diagnose cancers, design treatments, and then treat patients in an ever increasingly effective way. And we're going to hear that from both the clinical side, the research side, and from a patient as well. So thank you to everybody for joining us to speak today. Before I introduce the panel properly, I have the traditional housekeeping points. In the unlikely event of an emergency, please exit the lecture theatre and leave the Oglesby building. Who would have thought it? The fire assembly point is the bike shed, which I'm sure you all saw located to the left as you walk out of the building. So today we will be joined by three speakers. The first is Dr. Matthew Krebs, and I'm going to read this. He is clinical senior lecturer in experimental cancer medicine and Honorary consultant in Medical Oncology. Fantastic title. So Matt is here to talk about the really incredible target research project that is ongoing. This is really developing a new range of diagnostic tools to help design and identify specific cancer types simply through a blood test. And given the incredibly invasive and difficult procedures that a lot of patients have to go through just to get a biopsy, you know, the advances in this area are really exciting. And you may well have seen actually some of the news coverage in this area. Matt will be followed by Dr. Alicia-Marie Conway. I'm going to read this again. Alicia is academic clinical lecturer and cancer of the unknown primary sub-study lead. So cancer of the unknown primary is where a patient has a cancer that's been detected, but it's a secondary, and the usual diagnostic tests and scans, biopsies can't identify where the primary cancer has come from, and she's leading some research which is helping to identify the origin of those cancers again through blood tests. And finally, and absolutely not least, we'll be joined by Neil Osborne, a patient here at the Christie, who's going to share his journey and his experience of being on a clinical trial here. So with that, thank you again for joining us. There will be an opportunity for Q&A at the end, so please think of those questions and store them up ready for the Q&A session at the end. And with that, I will hand over to Matt.
Great, thank you. Thank you very much for the introduction. And thanks for the opportunity to speak at this event and tell you about the Target National Study, which is a really critical study that the Christie Charity is supporting. So really appreciate the opportunity to speak with you this morning. So this study is all about precision medicine. So essentially trying to get the right drugs, the right treatments for the right patient. And we hear lots of different terms. I'm going to give you a bit of background first of all, just to orientate you to what this is all about, and then I'll tell you about the study. So we hear these different terms, and the first one perhaps is stratified medicine. So we're sort of moving away from, I've got a pointer here hopefully, we're moving away from thinking about cancer as just lung cancer and treat everyone the same, or bowel cancer, treat everyone the same, to working out what's actually going on at an individual patient's level in their tumour. What's gone wrong? What are the faulty genes? What's happening that's driving their cancer? And we can do sort of what we call biomarker testing, or looking for faulty genes, let's say. And then we can stratify patients into different groups depending on the faulty genes that are present in their cancer, and then give very bespoke treatments directed against that. So that's what we mean by stratified medicine. And personalised medicine just means, of course, what's the right treatment for any individual person. So looking at that person as a whole, looking at their biology of their tumour and selecting the right treatment for them. So we use different terminology, but it all comes under this concept of precision medicine, understanding the biology of a cancer and selecting the right treatment rather than using sort of older style chemotherapy drugs, which obviously have limitations. So if we can do this, it's just some examples of what this might look like. There are some patients who have something called an EGFR mutation in their cancer, and we can give EGFR inhibitors. We can have something called BRAF mutations in cancer. We give BRAF inhibitors. We can have things called BRCA mutations. We give PARP inhibitors. That's just to illustrate the concept of different genetic mutations we can find in cancers, and then giving very bespoke drugs that have now been developed. And here's just an example of how effective this can be. So this is a lung cancer patient who's got disease that's spread to their brain. And I'll just orientate you. These sort of very white areas in the brain that you can see are areas of cancer. And this patient was found to have an EGFR mutation. It happens in about 15% of lung cancer patients. Went on to an EGFR inhibitor, and you can see just after a short period of time on the next scan, all those areas in the brain have shrunk down very nicely and are no longer visible. So this can be really effective if we get it right. Now, there are a whole raft of, you don't need to look at the detail of this, it's just to make the point, there are a whole raft of new drugs that have been developed over recent years targeted against these individual genetic mutations. And this actually stops at 2021, so it's quite old. There's a whole load in the last few years. And there are an additional, there are tens of new drugs in early phase trials looking at new targets. So these are developing all the time, these so-called precision medicines. And it's become increasingly important for us to be able to test for these faulty genes. So it's all very well having the medicines and being able to offer clinical trials, but if we can't do the test in the first place to look for the mutations, we'll never be able to select the right treatments. So it's become increasingly important that we look for these genetic faults in patients' cancers as a routine. Now, the way we've done that historically is through biopsies. So biopsies, for those who are not familiar, is when you would essentially try and put a needle into an area of the cancer and take a small sample away. So this is just an illustration of showing you lots of scans today. You can be expert in radiology scans. This is a patient's liver. The darker areas that you can see are areas of tumour within the liver. And under imaging, we can pop a needle into an area of cancer. Excuse me. We can take a small sample out. And it can be a tiny sample, a little dot that you can see there, that we put into formalin and put it into paraffin. And then we can do fancy tests to extract the DNA from that sample and run it on sequences that will tell us the DNA code and whether there's any fault in that DNA code. So that's historically how we've done things. But biopsies, as we've heard in the introduction, can be invasive. They're unpleasant for patients to go through sometimes, not always. The radiologists are very good at doing them, but still, it's having a needle put into an area of your body to take a sample. And there can be risks involved or complications. And when we come to looking at doing the genetic tests, when we see a patient in a clinic considering clinical trials, it may be their biopsy was done 4 or 5 years ago. So is it still relevant to what's going on right now? So an alternative is to use blood samples. Much easier to take, simple blood sample. And the technologies have evolved tremendously in the last 5 to 10 years, where we can now we can now extract DNA that has come from the cancer. It's circulating around in the bloodstream, and we can take a blood sample and extract those tiny, tiny fragments of DNA, and then run that on the DNA sequences and look for faulty genes through doing the blood test. And we call that circulating tumour DNA. So it's DNA that's come from the cancer and is circulating the bloodstream. So this is great. So we said, well, right, we've got a challenge. We need to be screening our patients for faulty genes so we can select the right precision medicines. How do we take this forward? How do we start offering this for our patients? And that's when we went to the Christie Charity to say, look, we would like to develop a study to offer this test, this state-of-the-art blood test for our patients, so we can try and get people onto precision medicines based on their individual cancers. So we actually originally did a pilot study quite a long time ago. It was between 2015 and 2020 that the Christie Charity funded. And we published this in Nature Medicine, which is one of the highest impact medical journals out there. And we proved the principle that we could do this. We could take a blood sample, we could do the test, we could get a result back, and we can get patients matched onto the right treatments through doing that. And because of the success of that, we wanted to roll this out nationally. So the charity has been very kind in supporting rolling this study out across the country. And essentially what we're doing, for patients who have already received some treatment and perhaps do not have any other options available, but are well enough and want to try a new treatment, will be referred through to one of the experimental cancer medicine centres around the country, of which Manchester is one, but there are 19, I think, in total, 18 or 19. And for patients presenting at those centres, we've opened this trial that allows then those doctors and nurses to take a blood sample from patients, so this circulating tumour DNA blood sample, do the analysis, and then get the results back within a couple of weeks. And we sit in a tumour board meeting, which I'll come back to in a second, and we look at what we found and whether there are any trials available around the country, because we know patients are willing to travel for the right treatment. So we look all around the country as to what might be available. So this is just an overview of where the study is up to. It's sponsored by the Christie NHS Trust. The funders are the Christie Charity, but also the Sir Bobby Robson Foundation in Newcastle. And the patients that we're recruiting are those that are referred through to an experimental cancer medicine centre for consideration of clinical trials. Now, we opened this in July 2021, so it's been running for 4 years. We've got 19 centres involved. We've now got over 4,000 patients recruited, and we can go up to a minimum of 6,000 and potentially more. And the study will run currently until January 2027. And because we're now offering this test, we're starting to attract industry to the UK. So companies that are developing new medicines against certain faulty genes want to be able to, of course, identify the right patients to put onto those trials and try those medicines. So we're attracting industry into the UK to place more and more opportunities for our patients, which of course is a great thing. We set up this molecular tumour board. So it's one thing taking the blood sample and then running it through our fancy tests. We use one of the market-leading commercial providers for the test, actually, Foundation Medicine. So we send a sample off for the testing. But when you get the result back, it's quite complicated. And actually, not all clinicians are used to looking at very complicated genomic data and how to interpret that. So we set up this educational forum, the Molecular Tumour Board, where any patients recruited to the target study, when the result comes back, we can discuss those cases through the tumour board. There's lots of expertise in the room, and we can talk through what the results mean and whether there might be a clinical trial available around the country for those patients. It's incredibly valuable. We all learn something new every meeting, and it's been recognised by the Royal College of Physicians for educational value as well. We've also built in, so we're doing this blood test and we're matching patients with clinical trials, and I'll show you some examples in just a moment. We've also built in sort of future-proofing for this. So whilst we're talking about one particular blood test now, there are going to be more blood tests available in the future, and we want to be at the forefront of that in developing those new tests and new ways of treating patients and new targets that we can develop drugs against. So I won't go into this in detail because we have a couple of so-called sub-studies that allow us to take additional samples from patients recruited to this trial so we can start to work up these new blood tests for the future. And Alicia's going to talk to us in a second talk around one of those particular tests, the CUP-COMP. So I won't say anything more about it just now. So here's just a couple of case studies. For example, a patient who was recruited in Leeds, a 67-year-old new grandfather diagnosed with colorectal cancer and had 2 lines of previous chemotherapy treatment. Well enough for a clinical trial, was referred through to the Experimental Cancer Medicine Centre, offered the target national study, had a blood sample taken, and identified something called an ATM mutation. And we had a specific trial for that, called the Atrium trial. The patient was recruited to that study and had 7 months of controlled disease, enjoyed time with their grandchild, before going on to a new standard of care treatment that had subsequently been approved in the time that they'd been on the clinical trial. So it opened up even further opportunities for that patient. A patient in Guy's at King's College, London, a 42-year-old mother of 2, I'm sorry, I have to read off my screen, apologies, who loves exercising, outdoors, cooking and eating Thai food, especially spicy food, important fact, was diagnosed with a cancer of unknown primary, which we'll hear about more in the next talk, while she was 23 weeks pregnant, but we're now with a 20-month-old daughter, and had received, again, 2 prior lines of chemotherapy treatment. Came to the Experimental Cancer Medicine Centre in London, was offered Target National, had a blood test, and was found to have something called a high tumour mutational burden. I won't go into the detail, but essentially that opened up an opportunity for immunotherapy for that patient when she had no other options available to her. And she's had, at the time of this case report being sent to us, had 8 cycles of treatment by that time, was feeling very well, had an excellent response to the treatment, and had quality time to spend, of course, with her daughter. So clearly was very appreciative of being able to take part in this for an opportunity that never would have arisen without the ability to do these genetic tests. So really, really critical. A final example, a patient recruited at the Christie, a 58-year-old mother, wife, dog owner and avid gardener with a healthy lifestyle was diagnosed with lung cancer. She'd actually had, I think, also 2 lines of previous chemotherapy. No other options, came across to our Experimental Cancer Medicine Centre, offered Target National, had a blood test, found to have a really rare mutation for lung cancer, something called NRAS. We had a specific trial available against that, and really she had a dramatic response to treatment. So again, some imaging. I might start asking questions now. What do these scans show? So this is this patient's lungs, this is the heart. These white areas scattered throughout the lungs are areas of cancer. The black actually is the normal air in the lungs. And you can see just after a couple of months on treatment, lungs are looking beautifully clear, so this is all normal. The white areas are blood vessels that you can see. It's these sort of round areas of tumour that have shrunk down very nicely and almost disappeared. And she was on treatment for a year before her cancer started to grow again. So again, an opportunity that would not have arisen without being able to offer the genetic tests. So where next? I'll sort of wrap up with what happens next with this. Now, the NHS does offer some genetic testing for all cancer patients, but it's relatively limited because it's only genetic tests that would allow for a treatment that's reimbursed by NICE or the Cancer Drugs Fund. But there's a wealth of other information available within those tests that could open up clinical trial opportunities that at the moment the NHS is unable to support reporting. So on the one side, we've got NHS routine with a limited number of genetic tests that are done, which of course are very valuable. But on the other side, we've got all these clinical trial opportunities, new options for patients for other genetic faulty genes that don't currently have a routine treatment available on the NHS. So Target National is filling this gap. We are able to offer this test to support enrolment into clinical trials. But really, where do we want to go with this? We need to transition this to routine care. That's the obvious thing. We want the NHS to be able to support doing this for all cancer patients who would be suitable, are well enough for clinical trials. So how are we doing that? we're working with something called the NHS England Circulating Biomarker Genomics Network of Excellence, mouthful. But essentially, it is just the route that we need to go through to try and get an application to the NHS to say, could the NHS please support this? Now, we're well engaged with them, and I think in principle they're supportive of this, but the issue, of course, always is funding. And I think it will be at least 2030 or beyond before we could get this test potentially reimbursed on the NHS. Even then, it's not a definite. So we're working hard on collecting data, recruiting more patients. We're looking at the health economics. And because this study will potentially end in 2027, we're actively thinking about what do we do in that gap from when this study ends to when it might be available routinely on the NHS. So we're currently thinking about ways we could transition the programme and funding opportunities to address that. So we don't lose this opportunity during those few years. Finally, just to acknowledge a huge team of people that are behind this. Running these clinical trials are an immense amount of work with lots of different people involved. This really is just a snapshot of people. I'll just give a call out and embarrass Eleanor Todd, who is the current main project manager for Target National and keeps all this going and keeps all the balls in the air. But we've got a trials unit, we've got a translational research team, we've got Foundation Medicine who we collaborate with. We've got our digital team who support with software that helps us link clinical data and genetic data and supports our tumour board discussions. We've got all the sites involved at 19 centres, all the nurses, doctors, admin teams who help deliver this. The Christie is a sponsor, and of course, not least, the funding behind this through Christie Charity and the Sir Bobby Robson Foundation, who are currently supporting this. So with that, I think I've gone over slightly, I apologise, but I shall end there. Thank you. I think questions are going to be at the end. So I'll hand you back to yourself, or I'll just hand over to Alicia for the next talk, who'll talk about CupComp sub-study.
Thanks, Matt. I'm delighted to talk to you about my research that I've been doing since about 2017. And it really, throughout it, what's underpinned it is access to blood samples, which has been enabled through the original Target study and Target National. So I'll talk a little bit. Some of it might be a bit technical, but I will talk you through it. So I did my PhD in 2017 at the Cancer Research UK National Biomarker Centre, and the focus of it was to try and improve the diagnosis and treatment stratification of patients with cancers of unknown primary. So as Bob introduced, it's actually a little known cancer entity. Not many people know about it, but essentially it's a group of cancers that are left over at the end of all the diagnostic process where we couldn't find where this advanced cancer started. So we have a biopsy, we have radiology, all the clinical information, and we're still not certain where it came from. It's classed as a rare cancer, but actually makes up 2% of all new cancer diagnoses. And unfortunately, because it's spread at diagnosis, it is the sixth leading cause of cancer death. Often patients have had vague symptoms, they might present to A&E unwell, and then have a multitude of diagnostic tests and no answer at the end of it. So it's a very challenging cancer type to research. It's also made up of lots of different cancers, so not each one cancer is the same, which makes it difficult to do research. And because patients have had multiple tests on their tissue biopsy, often there's not much left over for research or for the molecular profiling that Matt's described, to try and find those faulty genes that might help us direct therapy. There is a small subset of patients who have CUP where we think we might know where the cancer started. So one example is if a patient presents with lumps under the armpit, and if they're female, we might suspect this came from the breast. However, we can't find a tumour in the breast. But looking under the microscope, we say, it looks like a breast cancer, let's treat it as a breast cancer, and those patients do better, and they get access to breast cancer treatments. So that's this favourable subset over here, but it's only 20% of patients with CUP. So most patients fall into this unfavourable subset, which basically means we cannot decide what this cancer looks like, and they have access to chemotherapy, but it doesn't work very well in most patients. So the aim of my PhD was to try and see, because we haven't got much tissue, can we use blood tests to try and move more patients into this favourable subset? Can we molecularly say they look like this cancer type? And that's important because we need to choose the best therapies for patients. So Matt's already touched on looking for faulty genes using tumour DNA. So he's talked mainly about mutations. We actually were looking at a different type of genetic change. It's called DNA methylation. So I guess if mutations are a change in the coding, DNA methylation is attached to the DNA. And we describe it like a postcode. So the pattern across the DNA is your postcode of where the cancer came from. And each cancer type has its own postcode. So this is a plot looking at DNA methylation patterns. Each dot is a cancer. And when we look at the DNA methylation patterns, we see they all cluster in neighbourhoods together. So their postcode tells them what cancer it is. So that's a breast cancer cluster there. And then you've got brain tumours over here clustering away from each other. So we know that this is from the Cancer Genome Atlas. This is based on tissue. So we know in tissue, we can find out where the cancer came from their DNA methylation patterns. But tissue is the issue. We don't have enough tissue to do this test. So that brings us on to the blood tests. And as Matt introduced nicely, we know that in patients with cancer, we have DNA that's floating around in the blood that we can extract to try and look for the tumour signal. But it's very small amounts of DNA. And in that small amount of DNA, you've got your normal cell-free DNA, so that you would produce normally. And then you've got tiny bits of tumour DNA in blue. So you're trying to use small amounts of DNA and find an even smaller signal within that. So it's technically very, very challenging. And my PhD helped to develop an assay to try and use tiny amounts of the DNA that we've extracted from blood, enrich it for the methylation signal, so only capture those bits, fragments that are methylated, and sequence it to get the pattern, to get the postcode from the cfDNA. And then detect the tumour signal using machine learning algorithms. So artificial intelligence was needed to help us. So I won't go into the technicalities of the artificial intelligence, but it's called CUPID. We take all the publicly available tissue data from the Cancer Genome Atlas, which gives us all the postcodes of known tumour types, so 29 different tumour types. But we need to dilute that signal because the machine needs to be able to see a diluted tumour signal, like cfDNA. So we dilute all this tumour signal into lots and lots and lots of different dilutions to mimic the cfDNA, and then we can train the computer algorithm to look for a diluted tumour signal. So that's the CUPID assay in simplified terms. And then we thought it would work quite well. We didn't know how well. So using samples from patients where we do know where the cancer came from, that were recruited through the original target study, we applied the CUPID algorithm. So what we found is in 143 patients, we could make a tumour diagnosis in the majority of patients. So our sensitivity, we could detect in 85% of patients. And when we did make a prediction, we were almost always right. So we were correct 97% of the time. So we only got 4 incorrect cancer calls. So we were quite pleased that we had a fairly accurate CUPID algorithm that for most patients will give some sort of prediction. So the next thing was to try and see if it worked on patients with CUP. So again, patients recruited through the original target study or through our CUP biobank here at the Christie, where we applied the algorithm and we got some CUPID predictions, which is great, but how do we know if they're right in CUP? Because we have no ground truth, they haven't got a diagnosis. So some patients, 15 patients in this cohort, after lots of investigations and subsequent treatment during their cancer journey, we actually do work out where the cancer came from. Sometimes with additional tests like the mutation testing, we have more and more clues that tell us actually this is probably a lung cancer after all. So in the 10 patients, our CUPID prediction was exactly that, what the final diagnosis was for these patients. In other patients, we can narrow it down to maybe 2 or 3 cancer types that we suspect based on the radiology and the biopsy. So this is this section here where we say they're clinically consistent. So we don't know for sure, but the algorithm is making a prediction that we maybe suspected anyway. And then there's 6 patients down here at the bottom where we had no clue. We just didn't know where this cancer had started. And it was making predictions. Now, we don't know if those predictions are right, but those patients would have no other options if they didn't have a test like this. So we were pretty pleased in this small cohort that actually we were making reasonable predictions in CUP as well. And just going back to those 15 patients that finally got a diagnosis after a long diagnostic journey. This plot is each bar is a patient and their journey, and it starts from when they were referred to the Christie. So they also had a long diagnostic journey to get to the Christie, so that's not shown on this plot. And it's months along the bottom, and when it's the bar's grey, that's how long they were treated as a patient with cancers of unknown primary. And when it switches to a colour, that's when that patient got their diagnosis of cancer. And you'll see it's highly variable. Some patients were treated for months as a cancer of unknown primary before we finally worked out where the cancer came from. And on average, it was 7.1 months before we got the diagnosis for these patients. And this is without the test. So this is with just extra scans and extra biopsies. So what we hope is that if we could introduce a test like Cupid really early on in the diagnostic process, we would save all this time for these patients that actually they do have a cancer. We just don't have enough information to call it yet. So this is the point we're at the moment. We have the test and the Cupid algorithm, and we think it works really well with high accuracy in known tumour types. And it shows good concordance with clinical data for patients with CUP. But we've only tested 13 tumour types, and we know that sometimes the algorithm is good in some cancer types and it falls down a little bit on other cancer types, but we haven't tested it enough to sort of quantify that uncertainty. So the next step for this test is to test the test. So we have to, before we can ever hand a report to a clinician to say we think this is a lung cancer, we need to know exactly how well the test performs. And this is where the Target National CUP sub-study came from, because to test tests like this, we need hundreds of samples from patients with lots of different cancer types so that we can accurately say how confident we are when we make a prediction that it is what we think it is. So I've been really lucky that I've been really well supported by all of the Target National team to start taking extra blood tests as part of the CUP sub-study. So patients can consent to giving an extra blood test that we're banking and we're going to use as our validation cohort. So we'll get a breadth of cancer types because Target National is not specific to one tumour type. And we'll also get rarer cancers, which is really important because I think you may have heard of similar blood tests out there being tested at the moment, but they often focus on the really common diagnoses. And actually the rare cancers are left behind because they're hard to validate in, but also it's only small proportions of patients. So the big companies aren't going for the little rarer cancers, but actually we can actually focus this test to try and validate in rarer cancers if we have enough samples. So testing the test is the next step, and we've focused on tumour types we know are common in CUP cohorts and often missed. And then once we know the performance of this test, the plan in a few years' time would be to be designing a trial in patients with CUP where we deliver the test and change their diagnosis and change their treatment based on it. So this is all the work we're doing, and you'll see Target National Sub-Study is really key to the validation of it before we can get to the clinical trial where we can start saying this test works and we need to test it in patients with CUP. So I've got lots of people to thank, but we won't get this test into the clinic in months. It'll take years, and these blood tests are what help us to validate the assay.
Morning, everyone. Apologies in advance. My talk is far less technical than what's gone before. Just a few holiday snaps and my experience. So, just to kick off with a bit of background, I live in Northumberland with my partner Gillian and our golden retriever Millie. And in a former life, I was a director in a firm of chartered surveyors in Newcastle. And in fact, I'm still on the payroll. So my story begins really in late 2016, when looking at my computer screen at work, I noticed that I was finding it more and more difficult to focus on the text. So being a normal bloke, I just ignored it for 6 months, thought nothing of it. Eventually went to get an eye test at the end of 2016. And on the same afternoon, they referred me to the RVI. And by 9 o'clock in the evening, I'd been diagnosed with an ocular melanoma behind my right eye. So I was referred to Sheffield for treatment. And in early 2017, I went down to Chester and had proton beam treatments on my melanoma. And that was successful initially. The tumour was blitzed. And basically, they thought that it had been successful. But unfortunately, the tumour started to grow again. So in July 2017, I had my right eye removed. To be honest, I'd lost the sight in my right eye pretty much from the end of 2016. So it was probably the right course of action, and I should have done it earlier. But that's what happened. So I have a prosthetic eye in my right eye now. So in early 2017, I decided to finish work and just deal with the health issues going forward and basically live life to the full. Didn't know what the future held. I took inspiration from a lady who most of you will never have heard of, I don't think, a lady called Phoebe Schnetzinger, who was an American lady. And she basically, she took up bird watching, which is a passion of mine. I'll go on to that. In her 30s in America, and basically was very, very keen, traveling all over the world, whatever. And she got diagnosed with cancer 5 or 6 years down the line, I'm not sure exactly. And she just took the view that she wasn't going to have any treatment. She was just going to go for it. She had a lot of trips planned. She basically thought, I'm too busy enjoying myself to worry about cancer. So she actually carried on for 20 years bird watching and traveling. And the cancer didn't get her. She died in a car crash in Madagascar 20 odd years later, but maybe she was just lucky. So my passion for bird watching has been there since I was 12 years old. Some people call it an obsession. But later that became a travel and wanderlust thing. In 12 months, my partner and I took 12 months off and traveled around the world. We rented a house, took a sabbatical, went back to work afterwards, which was tricky. So basically, within 2 weeks of losing my right eye in 2017, I'd stopped work by that point. I decided that I was just going to go for it and, as I say, live life to the full. We went off to Namibia for a month, did a road trip through South Africa, Namibia, Botswana. 2 weeks later, I went off to Ghana for another couple of weeks bird watching. And between 2017 and 2022, I traveled extensively: Kenya, Philippines, Madagascar, Borneo, Indonesia, Canada. Spent all my savings, basically. But just decided that life was too short. In 2022, I decided to return to work as the 6 monthly scans that I'd been having for the first 5 years of that period had all been clear. So I went back 3 days a week to my old firm. But in the spring of 2023, those tests had actually gone from 6 monthly to 12 monthly. And on the 12 monthly test, the ultrasound scan, there was a cyst in my liver, and they knew that was there. But they identified that looked more solid this time, so they wanted to do further tests. So further tests followed, and unfortunately it transpired that the melanoma had metastasized to my liver. At this stage, the prognosis didn't look great, to be honest. The standard option or standard treatment is immunotherapy at the RVI in Newcastle, my local hospital. But we started to explore other options and look at the possibility of getting on a clinical trial. Unfortunately, there was a clinical trial in Newcastle which had recently come to an end, so I couldn't get on that one. I had to jump through lots and lots of hoops. Some of the stuff that these guys have been talking about, biopsies, and the original biopsy from my tumour was a number of years old. They were trying to see if that was going to be compatible with the treatment that they were going to be talking about on this trial. But to cut a long story short, and frankly, I don't understand all the technical stuff, I got on a phase 1 clinical trial at the Christie starting in January 2024. And I got to pay particular thanks to my partner, Gillian, who badgered lots and lots of people. So that trial is under Professor Thistlethwaite here at the Christie, and I'm now 18 months into that trial. So the drugs are 2 drugs combined, tebentafusp and PRAME. The first one of those drugs has now been approved, I believe, and the second one has been given a proper classification, so it looks like it might be going the same route. So I come down for weekly treatment. I travel 4 hours from Northumberland on the train. I catch a train on Tuesday morning. I have my treatment on a Tuesday afternoon. I stay over and I get the train back on a Wednesday morning, and it just becomes second nature, to be honest with you. There are some side effects, but they're not major. I had some pretty severe joint pain to start off with in my hips, but that passed after a few months. And now the main side effect is that I get a rash most weeks. I do sometimes take steroids if it's particularly bad, and I apply cream because it dries the skin. But to be honest with you, it's manageable. It's not an issue really. So the current position, 18 months in, is that there are now 2 tumours in my liver that are being measured. There were some smaller ones, but they're now deemed to be too small to be measured. There's been a 35% reduction in the size of those tumours from the start of the trial. And no new tumours at all. So I think everybody thinks that it's going reasonably well. Currently, thank you very much. Currently, I feel healthy and fit. So going back to 2020, I this trip in my mind, I wanted to go to China to do some bird watching in Sichuan, but COVID put a paid to that, and subsequently China basically shut down to outsiders for basically 3 years. So by the time of my diagnosis in 2023, the trip was firmly on the back burner and not looking at all likely, to be honest with you. All the staff at the Christie will know that I badgered them consistently throughout the first 12 months of my treatment, kept chipping away about a trip to China. I was rebuffed to start off with we don't really think it's a great idea you're going to Sichuan and when you're undergoing this treatment at the moment. But the treatment was going well and I felt healthy and fit. So other trips were possible during that first 12 months of the treatment. I went skiing, which is another passion of mine. We went to France in our motorhome. We went to Canada. We did sensible trips. I went skiing again in Austria this year. Anyway, eventually, after all that badgering, towards the end of 2024, I got the green light that they would consider that it would be okay for me to go on my China trip. So in May, June of this year, I've only been back a couple of weeks. We went to Sichuan for 3 weeks, which was pretty intense. Bird watching involves getting up at 4 o'clock in the morning and coming back at 7 o'clock at night, lots of hiking and whatever. And it all went really well. We had snow, we had rain, but the birds were amazing. It was an amazing experience. And I'm already planning my next trip to Eastern China in January 26th, and I've been given the green light to start planning that. So that's my next goal. What I would like to say is that this trial and the treatment has allowed me to continue to live my life normally, and I don't think that would have been the case without it, to be honest with you. So it's been incredibly important to me. I want to say a massive thank you to the team here at the Christie, Eilish, who's been my primary point of contact from day one and has been really, really supportive. Thank you very much. And the doctors, Gemma, Rachel, more latterly, who have encouraged me really to sort of continue with my plans to do further trips. I've got a few holiday snaps on here, which I'll bore you with. As I say, nothing technical. Here we go. So this is our trip to Sichuan, which getting a visa was reasonably straightforward, but a little bit sort of time consuming. But if anybody's not been to China and is contemplating it, it's an amazing place. It really is, and it's really sort of opened up for tourism. It's quite civilized. The hotels are nice. The food, if you like spicy food, is fantastic. The The Chinese have developed a huge giant panda reserve to the west of Chengdu, where we were sort of skirting around the edge. You can't get into most of it, but it really is a fantastic place. And these are just some of the birds that we were sort of targeting. So golden bush robin at the top. It's just Chinese monal on our first day in the snow. My starbird, which is a bird called Temminck's tragopan in the middle there, which took 8 hours staking out to get a look at that. Fire throats. Most of these are endemic birds to this part of the world, and this is Lady Amherst's pheasant. So we saw 300 species in 3 weeks. It was an amazing trip. So thank you very much for giving me the opportunity just to tell you about my experiences. But thank you.
Thank you, Neil, for sharing that with us, and thank you to all of our speakers. Q&A time. Who would like to ask the first question?
Just thinking about the stratified medicine. There's so many drugs there, which are presumably being given to smaller and smaller groups of people. So how does that work for drug companies producing those drugs? I mean, they're very, very expensive, or how does it work?
I'll just speak louder.
Yeah.
Thanks. Thanks for the question. You're right. From an industry perspective, companies would obviously want to focus on larger groups of patients when they're developing their medicines, and patients with rarer cancers have suffered because of that. So there are fewer options for rare cancer patients. We've not spoken about it today, but we have another program that addresses that exact issue of trying to offer some of these medicines to rarer cancer patients, because we can now do the testing through Target National, but it's access to those treatments. So we have a separate program that enables us to do that. And if that's successful, and we can show in really rare groups of patients, it might only be a handful of patients sometimes. We're in discussions with NHS around how could we get reimbursement and routine funding for future patients with actually very small amounts of data. And they're receptive to it. So that's still work in progress around how we'll achieve it. And the companies are supportive of us exploring that, because essentially we're helping to develop their drugs for them. So they will often offer the drug free of charge. They'll help support the trial that we're doing to do that, whilst they focus more on their commercial interest for larger groups of patients. So we work closely with pharma to try and achieve that.
Thank you, Matt. Any other questions? One here in the middle.
Thank you very much. Absolutely inspirational work. My question is about AI and how you think the ongoing developments around AI are going to continue to facilitate the speed of research and the sophistication of results and sort of the final, the final solutions really in terms of how people are treated.
Yeah, there's a lot of conversations around AI in healthcare, and I think AI used in the right way could definitely answer some of the big questions. I mean, when we talk about the genetic information that we're trying to glean, the amount of data that one person would have to go through, the useful information can only really happen quickly with AI. But there's whole programs to try and make it ethical and explainable, because there's this thing called the black box. You put data in, it comes out with a result, but what's that computer doing? And it is open to bias, and the way we use it and apply it needs to be done, especially in healthcare, in the right way. So I think it can answer the questions if it's applied properly, and part of our algorithm is trying to build in that explainability. So when it's wrong, why is it wrong? When it's right, why is it right? Rather than just trusting the computer says it's this.
Thank you.
I don't know whether this is your expertise, but triple-negative breast cancer, my understanding, there's no after treatment like there is with estrogen receptive. Is there any progress being made with that after you've had your chemo and you've finished everything? Would you know? Sorry.
So you're right. There's no sort of so-called maintenance treatment for triple-negative breast cancer. There are. I guess all I can say is there are lots of trials coming through trying to make advances for that particular subtype of breast cancer. There probably isn't something that immediately springs to mind that is going to be the next best thing right now, but there's a lot of activity from industry and academics to try and address that real unmet need for that patient group.
Could you tell me what impact on cancers has the immune system, if any?
Just tell me a little more. Are you thinking? What can the immune system recognise the cancer? Do you mean? Or okay, okay. So good question. Cancers are clever and can cloak themselves from the immune system. Very occasionally, actually, the example you gave about your colleague, friend, that never had any treatment for 20 years and was fine. Very occasionally, the immune system can just kick into place and identify a cancer and deal with it. And you do very, very occasionally see that, and it might be that's what happened for your friend. But there's been a lot of focus on immune system over the last decade or so, and there are now sort of so-called immunotherapy treatments, and the way to think about it is it essentially uncloaks the cancer, or kind of stimulates the immune system to recognise the cancer, and they can be very effective for some patients, not everybody. And we're still trying to work out the best biomarkers. So what things we've been talking about today, what can we measure in a patient's cancer that gives the best indication that they could benefit from immunotherapy? And there are some hints and some things that we look at, but we've still got work to do there.
Thanks. Do we ever envisage an affordable routine blood test which would identify a problem with somebody that was likely to become a cancer before it actually was a cancer?
So that's a very good question. So yeah, the search for, I guess, pre-cancer, or those patients at high risk of developing cancer. And I guess what we've talked about is trying to pick up the tumour signal in blood, but there is a whole additional research question about whether the signals in blood could from your normal cells could predict whether you're likely to develop cancer or not. And there is work to say that there are changes in your blood that might be able to predict cancer, but it's very early research. So there may be an application. Where I can see it being most useful is patients that already have maybe a family history or some other risk factor for a cancer, and they're the patients that you would monitor closely with blood tests to either detect early or maybe predict before it's arisen.
Any further questions? One here.
I'd just like to say a huge thank you for what you're doing.
Thank you, and I'd like to say back to you all, thank you for your support through the Christie Charity, because without that we would not be able to do what we do. So it's a huge enabler for us and obviously for cancer patients as a whole. So thank you.
And Matt, thank you for stealing my final line there. So it's wonderful. I'll just conclude by saying thank you again to Neil. We wish you all the best with your future birdwatching trips, and wish you many, many more. Alicia and Matt, thank you for all the work that you're doing with your studies, and we wish you all the best in getting success in those studies, but also then funding from the NHS. And thank you to everybody here for all the support you give to the Christie and the Christie Charity. I'm going to have to use the line again that Matt stole, and without which this sort of work could not happen. So you're making all of this possible. Thank you, and we look forward to seeing you at a future event.